My research paper is about using nanoparticles to alter DNA, and I study at Metlakatlan High School in Pennsylvania, and my interests are medicine, baking, dance, and tennis. My introduction, the title of my paper is Researching Methods That Use Nanoparticles to Change DNA Sequences. The main research question I set out to answer is how are nanoparticles more efficient in editing DNA sequences? And another question I wanted to research is how can nanoparticles be used with existing methods? For my literature review, some relevant research is when researchers at MIT use nanoparticles to deliver CRISPR technology to edit genes in mice. And an area of weakness in my research was that there was a limited amount of studies conducted that actually used nanoparticles to edit DNA. To analyze my data, I connected results from multiple sources to thoroughly explain the science and break down different types of nanoparticles that can be used to edit the DNA and how they can be used with existing technology such as CRISPR. And the results I found were that nanoparticles are a key method in editing DNA as they are more effective than other technologies and are very flexible in being used with existing devices. For my conclusion, my research question was how are nanoparticles more effective in editing DNA sequences? And my findings were that ionizable lipid nanoparticles are a promising method and at transporting siRNAs to edit DNA, and using carbon nanotubes to deliver DNA is more efficient than CRISPR technology, which is the technology that is currently being used the most. And my paper has contributed to a deeper understanding of how nanoparticles can work with existing technologies and how they can be used to change DNA. In my experience, I found that Dr. Ilova was very helpful in giving feedback and helping me just start my research paper because I didn't really know what topic to do at first, and she helped me kind of narrow it down. And Professor Virgil was very helpful in explaining how to write the paper and how to organize the different paragraphs. And I was kind of confused at first about how to cite my resources and how to structure my paragraphs, but he really explained it in a way that made me understand how to do that.
Excellent, excellent. A question for Vedika, Dr. Christina.
Yeah, I would like to hear your opinion on how do you see the future of this integration of nanoparticles in the gene editing? How do you think, in your opinion, that things will develop?
I think that nanoparticles in the future will be used with CRISPR technology because they can help. They're small and efficient in delivering the CRISPR technology to the DNA in ways that the CRISPR technology can't, because in the CRISPR technology, it makes mistakes often, and nanoparticles can help eliminate those mistakes to just make it more efficient to get to the DNA in the body and change it.
Thank you. Thank you. Great.
Fantastic. Great job, Vedika. 2 questions for Vedika, and we want you to take it further with a passion project, all right? And we want you to join us this fall, where we are helping students create passion projects out of their research. Either we're working with professors like Dr. Christina, either you can do deeper research, which is phase 2 research with Professor Christina, or you can create it into a passion project. And we also will be helping you get media attention for the work that you will be doing. We've been experimenting that and we've had great success. So please join our program or send us an email so that we can create a plan for you. Okay?